My name is Naila Lindsay, and my topic today is retroviruses, aka Retroviridae. Retroviruses are animal RNA viruses. It is considered to be a very large group. Retroviruses include HIV, which causes AIDS, and HIV enters through direct diffusion. Uh, the HIV infected T cells, which T cells are a type of white blood cells, and this prevents the body from being able to produce antibodies. And a common drug used to treat this is AZT. It is used against HIV and it helps to stop the reverse transcription by targeting the enzyme reverse transcriptase. So, retroviruses are enveloped single stranded viruses. As you can see here, I don't know if you can see it, but right around here, this is the outer envelope that surrounds the entire retrovirus. And this little dark blue area in here is the capsid, the inner capsid that surrounds the nucleic acid and enzymes. The capsid contains two copies of the same positive sense RNA molecule, and this is called a diploid virus. Retro commonly means backwards, which kind of describes what's going on. The backwards reverse transcriptase, which converts RNA into DNA and not the common DNA to RNA. So basically, the retrovirus attaches and enters the host cell outer envelope, and it fuses with the retrovirus and allows it to enter the host cell. So the retrovirus attaches to the outer envelope, and once it attaches to the outer envelope, it fuses with the retrovirus and allows it to enter the host cell. The capsid dissolves and releases the enzymes and nucleic acid of the retrovirus. So once it enters into the host cell, the capsid then dissolves and it releases the retrovirus enzymes and nucleic acid. Then reverse transcription transcribes the viral RNA into viral DNA. The proviral DNA can now be transcribed into mRNA. So the retrovirus has then created viral DNA for itself. So once it transcribes into the new viral RNA, once it transcribes the new viral RNA into viral DNA, it creates DNA for itself. The viral DNA fuses with the host cell. The DNA enters into the nucleus when it fuses, and along with integrase, which cuts open the DNA of the host cell, it integrates the DNA of the virus with the DNA of the host cell. And then provirus is created. Viral RNA is created each time the host cell transcribes into DNA. So each time the host cell transcribes into DNA, viral RNA is created. And this can happen more than once. It can happen, 1, 2, 3, 4, 5 times, however many times that it transcribes, then it can happen. Viral RNA attaches to a ribosome, and through a process called translation, it will translate the viral RNA into viral proteins. So once the proviral DNA is transcribed into mRNA, it is then translated into viral proteins. The viruses are released through the cell membrane. So once the viral RNA is transcribed into viral proteins, the viruses are then released through the cell membrane. And the proteins will assemble new viruses. Once it's translated into viral proteins, it assembles into new viruses. So eventually the viruses will go through chemical changes to become mature, because these cells are immature. They are immature viral cells, so they can go through chemical changes to become mature. It repeats the cycle. After all of this takes place, it repeats the cycle, and it then can infect other cells. So this can happen over and over again, and it affects many different cells. As with prophages, the provirus can stay in latent stage, in which it is replicated along the whole cell DNA, which causes no damage. It can stay in latent stage, and it's replicated, and it causes no damage. And this is a short version, a short summary of what retroviruses are and what takes place inside a retrovirus.